Hi there, welcome to this video. So I'm going to go through the exam related questions for PAG 8. Just be aware that if you're doing it early on in the course, some of the questions will be hard because they'll link to things that you haven't come across yet. Or if you're doing it later in the course, then it's a nice revision, links a few things together. But definitely listen to the video because the more you hear the things that you're not so sure about, the more they make sense. And it uses quite a different language in the exam questions about practicals compared to the exam questions about the content. So definitely worth going through these videos. Okay, the first question. The figure shows some of the apparatus used in an experiment investigating water potential in potato tuber tissue. Okay, so we've cut out some discs of potato. So when you've done this experiment, you might have done discs or you might have done something that looked more like cylinders like this, but it's the same idea. Then you've got your sucrose solution, which you probably have used in a boiling tube. The discs were placed in boiling tubes containing sucrose solutions of different concentrations. So you've probably done an experiment where you changed the concentration. The sucrose in this experiment is broken down by the enzyme, so the sucrose is the substrate. Try and get in the habit of annotating your exam questions like I do, trying to model it for you. The percentage change of mass was then calculated, so percentage change, you don't need to work it out, they've done it for you, but it's always going to be the difference divided by the original value, that means the first value, times by 100. Okay, make sure if it's getting heavier, it's a positive number. If it's getting lighter, you've got a negative number. So we've got our results just here. State 2 details of the procedure that must be followed to obtain valid results. Okay, so we need to know this set of evaluative words to do with experiments. So the word valid means that you're really answering the question. So what is the question that this experiment is trying to answer? So it's trying to ask you the question of how does, sorry, I don't know why that's jumping all over the place. How does, oh, it really is jumping all over. Sorry. Give the board a little wipe, perhaps. The board might not be very clean. Maybe that's the matter. Don't know. Let's do some undoing. So, this experiment is trying to work out, answer the question: how does the concentration of sucrose solution affect the change in mass of the potato? Okay, so this is always our independent variable. This is always our dependent variable. So, sorry, that's jumped all over the place. How does the, oh, what is it doing? How does the, wants it to go down there. How does the concentration of sucrose solution affect the change in mass of potato discs? Okay, so how can we be sure that these changes here that we've recorded are due to the change in sucrose solution and not due to another factor? That's what valid means. Okay, so we need to make sure that we're only changing the independent, so we're only changing the concentration, okay, and all other variables are being kept the same. Okay, so basically that's what valid is always going to mean. Is it how do we know that our results are answering the question? How do we know that our investigation is answering the question we set out to achieve? Okay, and to do that, you need to always change just the independent variable and keep all other variables the same. So we know that we're changing the concentration. They've told us about that. So the only thing we can really elaborate on is how are we keeping other things the same? And you need to try and basically put as much detail as you can. So, for example, we could talk about the discs. So all of those discs that we're using need to be the same. Now, you can't just say keep the discs the same. Okay, that is not going to be enough for a mark. So an ideal thing that you could say is they need to have the same surface area. So if you've cut them, sorry, just surface area. If you've cut the discs to the same size, so for example, if your discs, rather than cylinders or something like that, if all of them have the same thickness and all of them have the same diameter, then they will all have the same surface area, won't they? Yeah. So try and elaborate, try and use your language to make your answer worth a mark. Just using the same discs is not enough. You could talk about using the same variety of potato. You could talk about always removing the skin. You don't want some with skin and some without the skin because it'll be different. But you could talk about removing the excess water on the surface before reweighing. You only want to measure the mass of the cells. You don't want to be measuring any water that's sticking onto the edge of the cylinder or of the disc. Okay. You could talk about always using the same number of discs each time. So always use 5 discs in each experiment. You could talk about using the same volume of solution. You could talk about keeping the temperature the same. Or you could talk about covering the tubes to prevent evaporation of water. Okay, you only needed 2 of those. Now, on this mark scheme, they didn't want any numbers, but sometimes they do. So sometimes it's worth, for example, if you're talking about same temperature, you could say do them all at 21 degrees C, room temperature. Yeah. If you're talking about same volume of solution, you could say 20 centimeters cubed. So anything that is a measure quantity, put what it's going to be, what the quantity could be. Sorry, get rid of that. Just to make sure. The other thing you can do if you want to really make sure you get the mark is say how. So things like temperature, how do you keep that the same? You could say that you'd use a water bath. Yeah. If you're talking about the same volume of the solution, you could say you'd use a pipette to measure it. If you're talking about the same surface area, we've said measure it to the same diameter and the same thickness to give the same surface area. So some of them you can elaborate on more, and some mark schemes will be pickier than others about the detail. Second one, explain how a student could use the data in the table to determine the water potential of the potato tuber in the tissue. So hopefully you did this as part of your practical. So basically, what we need to do for this one is you need to plot a graph. So for this experiment, hopefully you're familiar with the idea that you plot a graph that looks like this. So you've got your percentage change of mass on your y-axis, and you've got your concentration of sucrose on your x-axis, and you plot the points, and what you find is that they, at some point, they go through 0. Okay, whatever your line looks like. So at some point, they're going to go through 0. And what we're looking for is that intercept where the line crosses 0, the point where it didn't get heavier or lighter. Okay, so what you need to do is say that you would plot a graph of what? So of percentage change of mass against sucrose concentration. Okay, then you need to say that you would find the concentration of sucrose that gives no change in mass, so where it's on 0 on the percentage change line. So find the concentration of sucrose where percentage change is 0. Okay, so that's that big point there of drawn in red. You can always do a little sketch graph to show exactly what you mean. That would be absolutely fine. Then that value there you look up in a lookup table. So you look up the concentration in a lookup table to find the water potential. That's the water potential symbol just there. Hopefully you're familiar with that. Okay, so that's probably the method you've looked at. There is another way of doing it, which is in the mark scheme if you want to have a look at the paper copy as well. On to question 2. A student carried out an investigation into the effect of different concentrations of sucrose on tissue from different vegetables. 4 different vegetables were cut into slices. The slices were placed into solutions containing different concentrations of sucrose. The change in mass of the slices was measured after a set period of time. The results are shown. Okay, so very, very similar, but this time instead of just potato, they've used various vegetables. The student made an error with one calculation from the potato samples. Calculate the correct value and show your working. So we're looking for a mistake in the potato section. Okay, so first of all, we're looking for which one of these results here that she's calculated are going to be wrong. Okay, so you might want to just work them out. So we're looking at the mass at the start, mass at the end. So if you do one of those minus the other, you can find the difference. Okay, and then change a mass as a percentage is going to be the difference divided by the original times 100. So if you work each of those out, what you find is that this one here is the one that's wrong. Okay. So first of all, it's identifying which one's wrong, and then it's showing you're working out. So we should have worked it out from these values here. So in order to find the difference divided by original times 100, so the difference is 3.44 minus 2.78. Okay, that's your difference. Divided by the original one, so divided by the lower one, 2.78. And then times 100. So that's your working out that you should have shown. And then you should find that that equals minus 19.19. Okay, so minus 28.77 was wrong. All right, so it's just a case of knowing how to work out percentage change. So you don't just need to work it out for your PAG, you need to actually be able to do it for the exam as well. They might ask you questions about it. So minus 19.19. Then it says estimate the concentration of sucrose in the cytosol of Swede cells. So we haven't got a graph this time to use, so we're just trying to look where there was no kind of gain or loss. Okay, so we're looking for where is it going to cross zero? Where would the percentage change be zero? So we're looking for Swede. So we're looking in this section here. Now we haven't got a zero, have we? So we're trying to say that it would be somewhere in between these. Somewhere in between these two. Okay, so if it's those two, if we go across, that one is the result for concentration of 1.0, and that one is the concentration for a result of 1.5. So we don't know exactly what it is, but we know it's between a concentration of 1 and 1.5. So if we go back down to the space where we're writing our answers. So it's going to be between 1.0 and 1.5. You probably wouldn't write that. You would probably estimate a value, and as long as your value was in between those, then it's good. All right. You need to write units as well. So concentration of sucrose is moles per decimeter cubed. Okay. So you could write those units, moles per decimeter cubed. Okay, or more scientific way to write it, instead of using the forward slash for the per, you put a minus up there, so to the -3. Okay, that's a more scientific way of writing it. Next one, what other factor will change as the concentration of sucrose solutions change? So hopefully we know that if we have pure water, so if we've got distilled water, it's 100% water, then the water potential will be 0. And hopefully we know that if we add solutes, then it's not 100% water anymore, it's less than 100% water, and therefore it will have a negative value of water potential. Yeah, so it might be, e.g., minus 250. Yeah, kilopascals it will be. Now, that's not what they want you to write, that's just me explaining it. What factor will change is the water potential. All right, it will change the water potential. Water potential is affected by the amount of solutes, concentration of solutes that you add. Part B, describe how the student should represent the data from table 20 as a graph and explain why this is the correct way to represent this data. So hopefully you've drawn a graph in your PAG and you know how to do it. So we call that type of graph a line graph, first of all. And the reason it's a line graph is because both variables are continuous. Yeah, they're on number scales, aren't they? If one of them's discontinuous, then we would do a bar graph. Okay, so hopefully we know that the graph looks like this. It's asking us to describe it really, rather than draw it. So you can always draw it as a prompt. Yeah, so we could say on the x-axis here, we've got our sucrose solution, sucrose concentration. Always good to put units. They haven't put them on this mark scheme, but it's a good habit to get into. So moles, decimeter cube. And then on the y-axis, percentage change of what? I always simplify it to percentage change, but percentage change of what? Can't just say percentage change. That's me just saving time. So of the mass of the potato. Okay. The x-axis, why does sucrose concentration go there? So we can say that that's because it's the independent variable. Independent always goes on there, and dependent always goes at the side. So why does that go on the y-axis? Because it's the dependent variable. Okay. And then because this one is from multiple vegetables, you're going to plot all the lines on one graph. How are you going to tell them apart? You could use different colours. Yeah, whenever we need to plot more than one line on a graph, we always add a key and use different colours. So hopefully that's nice and familiar. It's just getting in the habit of how to explain it. Okay, so by doing the PAG, you learn how to do the graph, but by going through these types of questions, you learn how to describe them, how to answer the questions about it. So in part C, considering the data in the table 20 suggests 3 improvements to the design of the experiment. For each improvement, explain how it will increase the validity of the data collected. So here's the same word they're using again. So validity, is it answering the question? So the question here is how is the independent variable affecting the dependent? So the effect of the concentration of sucrose on the percentage change of the potato. Yeah, so we need to be sure that we're only changing the independent variable, which is the concentration of sucrose, and we need to make sure that everything else is the same. So all, oops, it's a bit messy, even messier than normal. So all other variables are kept constant. All right. So we've got to try and improve our experiment. So if we go back up to our experiment and we'll have a look. Okay. Okay. So first of all, we could talk about, if we just say, it says they cut them into slices. So first of all, there's our first kind of criticism. They need to have the same surface area, don't they? So when they cut the slices, they need to be the same diameter and they need to have the same thickness so that they have the same surface area, because surface area will affect the rate of osmosis. So if we go back down, so improvement 1, we could say keep the same surface area of potato discs. Explanation to reduce the effect of surface area on rate of osmosis. Okay, let's have a look what else we've got. We haven't got much method on this one to go on, so we're going to have to use the results table as well to interpret what they did. So in the method, there's nothing else really we can criticise. So in terms of results table, what could they have done better? They could have done some repeats. Okay, so by repeating it, think about how many times you would repeat. So we usually say 3 repeats. Okay, and that's so that we can see if any of our results are anomalies. Okay, so if you got 2 that were similar and 1 that was different, then you would know that one's an anomaly. If you only do it once, you don't know if it's an anomaly or not. Okay, so we could say repeat 3 times. So they've done it once, so we could say twice more. And that would enable you to identify anomalies. Okay, you could say that it would increase repeatability as well. All right. The other thing is, the whole point of this experiment is that we're trying to find where the change in mass is 0. So you either do that with a graph of where it crosses the x-axis, or we're looking at a table for where the percentage change is 0. Now, as we saw when we were looking it up for the earlier question, we saw that it was somewhere between 1 and 1.5. So if they'd done more intermediate values around there, we could be more sure of our answer, couldn't we? We could be more sure about the effect of the concentration of sucrose on the water potential, because we'd be more sure of what the water potential was or where it was 0, whether the percentage change was 0, sorry, and therefore we could look up on the lookup table what the water potential was. So more intermediate values of sucrose solution. It doesn't say this in the mark scheme, but generally it's always a good idea to use numbers. So more intermediate, so we could say here between 1 to 1.5, because we know that from an earlier question, don't we? Moles per decimeter cubed. And why would we do that? So that we can identify solute concentration, which is the sucrose concentration, with 0% change in mass more accurately. All right. Now you can see how similar these questions are, can't you? These related questions. They're very, very similar, and they're using the same language and the same ideas. Next one, a group of students investigated the effect of temperature on the membranes of beetroot cells. Okay, so this time we're changing the temperature, we're looking at beetroot. So hopefully this is another experiment you've done that's familiar. A colorimeter was used to measure the concentration of purple betulin pigment that leaked out of cells when they were exposed to different temperatures. So hopefully we're aware of our plant cell with our cell wall, with our cell membrane, with our vacuole, and it's in that vacuole there that we've got the pigment. So we can say contains betulin. We've got the vacuole membrane, which we call the tonoplast. Okay, and we've got our cell surface membrane here. So what happens in this experiment is that the pigment, I don't know if I got purple, let's see if I got purple, there we go. The purple pigment from here leaks out, so it's going through 2 membranes, the tonoplast and the cell surface membrane. Okay, and as we increase the temperature, moving down, hopefully we can say that the absorbance is increasing. So that means more light is being absorbed because the colour's getting darker. So we've got a darker colour there, absorbing more light. We've got a lighter pigment here, absorbing less light, which means less pigment was released up here, hence the colour's lighter. Released, whereas down here we got more pigment released. Okay, table 23 shows a summary of the data collected. Hopefully that's familiar. Then we've got a standard deviation in the last column. We'll talk about that a bit more later on. So our first question is, using the student's t-test formula below, calculate the value of t. So as I said at the start, depending on where you are in your course, you may not or may not have done the t-test, but listen to this bit anyway, okay, because the more times we go over it, the easier it becomes. So first of all, you should get given the equation, okay? Looks like a big scary equation, but you are given the equation. So the first thing you're going to do is you're going to use x bar A minus x bar B. Now, x bar, x with a bar on it, just means mean, okay? I know it looks fancy and scary, but x with a little line above it is called x bar. X bar is how we say it. Sorry, that's a bit of a blurry pen. So we call it x bar, and it just means the mean. All right. So that's harder than it easier than it looks. Then what we're going to do is we're going to use s squared from sample from A and s squared from B. Okay, so S is simply our standard deviation, so that goes to here. Let's put our S up there. And then you're going to divide it by n of set A and n of set B. Sorry, these pop-ups keep coming up. And that just means the number, okay, the number of samples we got. All right, so it does even tell you that down here, I don't know where x bar's gone off this, but it should say x bar is the mean, s is the standard deviation, and n is the number of readings. Okay, so they even give you a little clue there, which is a huge help. So you don't have to know the equation, and they're likely to even give you a little bit of a clue about reminding you what the letters are. Okay, so using our numbers from our table, let's just scroll up a little bit so you can see them. So this here is the mean, okay? So when we're looking for x bar, when we're looking for our mean values, our yellow ones, this is our mean result here. All right, we've got that already labelled up. So first of all, we want x bar A minus x bar B. Okay, so that's just the difference between the two means. It said we wanted between 50 and 60, so we're using this set of data in these two rows here. So you're simply going to do 0.44 minus 0.21 for your difference in your means. Then you're going to do that divided by square root of the difference between the two standard deviations divided by the number of samples that we're going to use. Okay, so I'm just going down on the mark scheme just to double check. I'm just telling you where the marks sort of belong. Okay, so looking at our numbers here, so our first standard deviation is 0.06. So we pop that in there. Let me scroll down a bit because you only need those bottom values. So 0.06, you see it's squared just there, and it's divided by the number of samples. Now it told you here the number of samples, number of readings was 10. So if I highlight that the same colour as well, to make sure it all lines up. So we're going to divide it by 10. Sorry, this should be right down here. it's the square root of all of this. Get rid of that. And then you're going to do plus. Standard deviation of your next one is 0.18 squared divided by number of samples, 10. Okay, and all you need to do is put all of that into your calculator in order to get your final result. So you can do it in stages if needed, if you don't know how to do it all in one go in your calculator, and you should get a result of 3.83. I know that's new to lots of you, but try and follow it through. And if we do one in each of these practicals booklets, you'll be an expert by the time you get to that stage in the course. So whenever we do statistics tests, we're then comparing them to something called critical values, because what we're trying to do in statistics is we're trying to work out, are our results statistically significant or not. And the way we do that is we calculate a number, which is like what we just did for that student t-test, and then we need to look it up or compare it to a critical value. Sometimes it's in a table or sometimes they just give it to you. So here they've told you that the critical value for T at a significance level of 5%, so we're 95% sure it's not due to chance. There's always an element that it is due to chance in biology. 18 degrees of freedom is 2.10. So at the moment, don't worry too much about all that in between stuff. The critical value is 2.10. Now, what we're doing with statistics is we're basically saying, is our result higher or lower than the critical value? Okay, so the value that we calculated in our previous one was 3.83. So we said T is 3.83. So we're going to compare our result to the critical value. So we've got T equals 3.83. Okay, we've got to say whether it's larger or smaller than the critical value. So 3 is obviously higher than 2. So we can say this is higher than the critical value. And then the last bit is interpreting that, and this is something you just have to learn. Okay, so if your value is higher than the critical value, then we say that we reject the null hypothesis. Now, the null hypothesis, null is like none, 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 no, nothing, isn't it? Null. Yeah. So the null hypothesis is that we're saying that whatever our independent value did not have a significant impact on the results. So we're saying temperature did not cause those changes in results. Okay, it's like the negative version of your hypothesis, saying that it did not have an impact. If our t result is higher, our t value is higher than the critical value, we reject that null hypothesis. So we're saying that actually the difference is significance. So the difference between those two results, was it 50 and 60 degrees, is significant. And it's using really specific language. So we always use this exact language. And there's almost a script you can follow for these questions. So the difference between those two results of 50 degrees and 60 degrees is significant or not due to chance. So the null hypothesis is saying the difference between those two values is not significant. It's just due to random chance. It could have happened for any reason. Whereas we're saying our value is higher than the critical value, so the difference between those results is significant. Our experiment has proved something. It's like the positive outcome of that. Okay. Plot B. The students plotted the data onto a graph, which is shown in figure 2.3. Describe and explain the pattern of data on the graph as the temperature increases. So we've got two different things here. We've got describe and we've got explain, which mean two very different things in terms of graphs. So first of all, in terms of describing, we can say that as we always start with the x-axis, so as the temperature increased, the absorbance increased very slowly, and then had a steep increase, for example. It would also be better, it doesn't say it in this mark scheme, but it would be better if we reference numbers. So we could say it increased very slowly. We could say until 30 degrees C, up until 0.06 arbitrary units, and then a steep increase, we could say up until 60 degrees C, up until 0.5 arbitrary units. Yeah, so it is a good practice to put numbers in, even though they haven't asked for them here. The explain, on the other hand, means why did that happen? What was happening to make that increase? So, to increase the absorbance, basically the way the colorimeter works is you put your sample in this little thing called a cuvette, and the colorimeter tries to shine a light through it. Now, if it's very pale, most of the light will go through and the absorbance will be very, very low. As the colour gets darker, okay, as the temperature increased, the colour got darker because more pigment came out of the cells, less light goes through, and therefore the absorbance is higher. All right. So what we can say is that there's an increase in pigment increases absorbance. And let's go through it step by step. So why did it increase very slowly at the start? So that must be because the membrane wasn't damaged at the start when it was cold. Oh, this part's annoying, isn't it? Okay, and then as you increase the temperature, think about what happened and why more pigment came out. So we could say the membrane became more permeable. You could say that proteins denature. You could say the phospholipids move further apart. Okay, there's lots of ways you can explain why, but basically, as you increase the temperature, membrane becomes more permeable because of these things, and therefore more pigment leaks out. The colour comes darker, and therefore the absorbance increases. All right. If you've done your questions already, just make sure you didn't put anything like enzymes denature because they are proteins, not enzymes. They won't let you have enzymes. Next one, question 4A. A student investigated the effects of different solutions on pieces of potato tissue. Back to potatoes. 6 potato rods prepared with a cork borer. Hopefully you know what a cork borer is. It's that little metal thing. It looks something like this. Oops. Oh dear. What? It must be the way I'm leaning on the pad or something. So it looks something like this, okay, and you push it through the potato. You can see what I'm trying to do. Push it through the potato and it cuts the cylinder, the cork borer. The student trimmed them to a length of exactly 5 centimetres. After treatment, 6 rods were placed in test tubes, submerged in either sucrose solution or distilled water. Okay, so they didn't do lots of concentrations, they just did sucrose or water. The treatment and liquid added to potato rod is shown in the table. After 30 minutes, the rods were removed from the tubes, the lengths measured. The student recorded the results on a piece of scrap paper. In the space below, present the student's results in appropriate format. Okay, so hopefully you're starting to get the hang of how strict they are on results tables, and you have to be able to do them without any guidance to get them ticked off on your PAG, but also they might ask you to do tables with no support in um, an exam as well. So it's really, really important that you know how to draw a table correctly, meeting all of their criteria. Okay, so the first thing to identify is your variables. So you need to make sure that you've got your independent in the first column. So I'll just do it to the side here as a general rule. So you're always going to put in your first column independent with your units. Now, most of the time, your second column is going to be dependent with your units at the top, and then some way or other, you're going to need to subdivide your dependent. I don't know how many sections in this one, and draw another line across, and then we finish off our table down here. And it's really important that it's all lined with a ruler. You need to bring a pencil and a ruler into your exam. That this drawing around the edge is a rectangle, that's not got this corner cut out, which lots of you like to do. That you've got units, that you've only got units in the heading, not in all the other boxes, that your decimals are the same, etc., etc. So first of all, identifying what our independent variable was, first of all. So, what was it that they are changing? Okay, what is their independent variable, and then what is their dependent variable? What is it that they were measuring? Okay, try and look it on the mask. Here we go, found it. Okay, so the first thing is they've changed the treatment. So this column here is basically our independent variable, what treatment they had. Okay, um, so you could describe what treatment they had. I suppose it's both of those, really, isn't it? Yeah, so you could describe them, or you could just say the letters. Either would be fine. And then the dependent variable is actually down here. It's their results that they found here. So these are the dependents. All right. So for your table, your independent unit is the different treatment of potato. Doesn't really have any units because they did various different things. All right, but we would draw that then like that. You could list all the different treatments, or you could say that there's like a key, and then we just put the letters there. Either would be fine. Um, then they measured the mass. So here they've got the final length, but they haven't recorded the start length. But they did say the start length right at the start here. So they trimmed them to a length of exactly 5 centimeters. So we could put length as our dependent length of potato, and you might want to put something like start, end, difference, change, yeah, measured in centimeters. For this experiment, we did do percentage change, so you might have added a percentage change column on the right hand side. You wouldn't have got penalised, but it did just say record their results, so you didn't need it. You must have this rectangle around the outside. You mustn't leave it without that. Okay. And then you needed to have actually put the results in. Please use a ruler. Excuse me, being lazy. Take me so long with the ruler tool on here. You'll make the video even longer. So they all started at 5. You're not actually penalised if you didn't put the start in because they all started at the same. And then your final ones, your end lengths, you just needed to copy from here. So we had 5, 5.1, 4.9, 5, 4.7, 5.3. Now you didn't need to put the units in there. Okay, units are in the heading. You don't need to put them in every one. However, they all need a decimal place. So these need to be 0.0, that needs to be 0.0, all these need to be 0.0. Decimal places need to all be the same. You might have done difference, in which case you need to work these out. Can you see how I always put the decimals? So the decimals are always consistent. All right, something along those lines. So make sure you've labelled your headings. Make sure you've got your columns the right way around. If you've got your dependent on the left, you won't get the marks. So make sure you know how to do tables. Hopefully you're having lots of practice in class. Part B, state how the student could reduce the uncertainty of their data. This question is always going to be the same, okay? If you get a question about uncertainty, there's no guessing. You've got to know what uncertainty questions actually mean. So this is always going to be about using more accurate or more precise apparatus, okay? So what were they measuring? They were measuring length. Yeah, and they were measuring them really small amounts because the potatoes aren't going to gain in size that much. They're measuring very small amounts, like 0.3 millimeters or centimeters. So very tiny amounts. Using a ruler is probably not the best piece of apparatus to use that. So a good piece of equipment that they could have used is calipers. If you don't know what calipers are, just Google it now. Just pause the video and Google it. It's just a little tool that you can use to measure the length of something. More precisely, so it'll reduce the uncertainty. Part C, explain how the treatment results in the explain how the treatment results in difference in the final lengths of rod A and E. So we need to look back at what A and E were. So for A, they boiled for 5 minutes and they had 1 molar of sucrose solution. And for E, let's highlight the ones we're talking about. I'm just going to do my answer here. Obviously, you can do them in the right space, but I'm just going to do my answer here because you can't see the table and my marking space at the same time. So they're specifically asking you, why did we get the difference in these two here? So the first one was boiled. So obviously, we've got to talk about what boiling is going to do. So boiling is going to damage the membrane, isn't it? Membrane of each cell. Remember, that little potato disc or cylinder is going to be made of lots and lots of cells. So it's going to damage the membrane of all of those cells. You could have said some kind of detail, like it denatures the proteins in the membrane or disrupts or destroys the membrane, the protein, something like that. And therefore, there will be no osmosis of water out of A. So if we look what happened to A then, A had a final length of 5, so it started at 5 and it finished at 5. Yeah, so it didn't change size. So we could say length stayed the same. What's it doing? Length stayed the same. That's right here. There, because there was no osmosis. Right. The third one was untreated. It wasn't boiled, so therefore, obviously, the membrane wasn't damaged. The proteins weren't denatured because they didn't boil it, and the final length was 4.7. So it's got lighter. So water has moved out by osmosis. Okay, so it got lighter. Water moved out by osmosis. All right, you write yours in the proper answer space. I'm just doing it on there because I couldn't see everything at once on the screen. Lovely. We'll get quicker because the questions become so similar. Explain how the treatment results in the difference between the final lengths of rods D and F. Next. Okay, so D and F. So we can see that D stayed the same and F got heavier. So D stayed the same, F got heavier. See how I annotate my work all the time? Try and get in the habit of doing that. So it was soaked in ethanol. So what do we think ethanol did to our membrane? Well, ethanol, we need to know, dissolves the phospholipids. Okay, that is something you need to know. Sometimes the versions of this experiment are temperature, and sometimes the versions of this experiment are alcohol. We need to know both. So it dissolves the phospholipids. And therefore there's no osmosis, and it stayed the same mass. This one was untreated, so we didn't damage the membrane, and that's why it got heavier. Explain why it got heavier: because water moved in by osmosis. Okay, so it's really explaining why things happen. So yes, it got heavier, but why did it get heavier? What was actually happening? Next question. Number 5. Water moves by osmosis in living organisms. A group of students used the following method to investigate osmosis in plant cells. They cut pieces of plant material of equal surface area, ensuring no skin was present. They rinsed it to remove cell debris. They gently patted the plant pieces dry with a potato towel. Oh, sorry, a paper towel. Potatoes on the mind. They weighed each piece and recorded the mass. They put the plant piece in 200 centimetre cube beaker. Should we start to draw it? It's nice to visualise it, isn't it? So here's our piece of plant. They covered the plant piece with a sucrose solution. They used sucrose solutions of various concentrations. They left it for 24 hours. They removed the piece of plant material. They dried it with a paper towel. They weighed the plant piece and recorded the mass and calculated the percentage change. Repeated twice more. Okay, so they investigated carrot, courgette, and potato. So hopefully all this is nice and familiar. Oh, let me go down. Yeah, the results are shown in the table. Nice and familiar, very similar to the earlier question. Explain why it was necessary to calculate percentage change in mass. So why didn't they just calculate the change in mass? Why did they calculate the percentage change in mass? Now, this is because they probably had different starting masses, which means they've made of a different number of cells, and therefore it's not fair to just say it increased by this many grams or that many got lighter by this many grams. Okay, that wouldn't be valid because there's another variable, the starting mass. So percentage change is much better. And it's always going to be that they had different starting masses. I've seen that question so many times. Okay. So this then allows a comparison between pieces of different mass. Okay, the whole fair test thing has been taken out of all specifications and mark schemes, so you can't say because it wouldn't be fair to. It allows a comparison to. You've got to learn the language they want you to use. The students identified replicate 3 of the potato in 0.7 as anomalous. Suggest a practical error by the students that might have caused this result to be anomalous and explain the likely effect of this error. So, replicate 3, 0.7. What vegetable was it? Forgotten already. Potato. Right, potato, 0.7, replicate 3. Okay, so it's the lowest. I got 6.1, 5.9, and 5.1. So why might they have got that low reading? What might be the reason for that lower reading that's an anomaly? Three of the potato in the sequence. Let me check that. And the mark scheme's saying that it's heavier than the other pieces, whereas I just looked up it was lighter. Replicate 3, potato 0.7. Potato 0.7. Replicate 3. Oh yes, of course it's a negative number. Let me look at, they're not the masses, are they? What are those numbers? Percentage changes. They're all percentage changes. That makes sense. So this one is a percentage change. Okay. So they've all got lighter. That's that's gone down by 6%. That's gone down by 5.9%. That's only gone down by 5.1%. So it's heavier. It's not got as light as the others did. Okay, that's fine. Always look at the headings. That's the trouble using a computer. You can't get it all on the screen. Okay, have I gone by? There we go. So it was heavier than the others. Other repeats as the percentage change was less. Okay, so why might it still be heavier than the others? What might they have done wrong? They might have not dried it properly. Okay, because you're meant to dry the liquid off the surface so that you're only measuring the liquid in the cells. So it might not have been dried enough. It might have been from a different part of the plant, different part of the potato. We should be using all cylinders from the same part of the potato because there might be different water potentials at the start, different parts of the potato. Okay, not dried enough would have meant that it's heavier, or different part of the potato might have had a different water potential at the start. All right, so why do they get an anomaly? Use table 16 to identify which plant cells contain the highest concentration of sucrose. Justify your answer. Okay. So, what we're looking for on here is we can use the means. Okay, we're trying to find out where it would intercept 0 on the graph. So where has it got a percentage change of 0? So for carrot, it's between there and there. For courgette, the crossing of 0 is going to be between here and here. For courgette. And for potato, the crossing of 0 is going to be between here and here. So we're after which one is the highest. So they're all a range, aren't they? Which is makes it difficult. Just reading the mark scheme just to double check. Okay. Okay, so we're trying to look for where the 0 is, basically, where it crosses 0, and we'd be able to see it much clearer if we drew a graph. Now, interestingly, if you have a look on the mark scheme, they have drawn the graph, but it doesn't ask you to draw a graph, it asks you to use the table. So it's going to cross 0 somewhere between 0.3 and 0.5 for the carrot. It's going to cross 0 somewhere between 0.3 and 0.5 for the courgette. And it's going to cross 0 somewhere between this one, 0.1, and 0.3 for the potato. So it asks you for where is highest. Okay, so the intercept is going to be higher on courgette or carrot than it is on potato. So that's the first thing that you can say. I'll just write my answer up here so we can see the results at the same time. Okay, so we can say that the percentage change. Oh, it's doing the silly thing. the percentage, the percentage change or the intercept is 0.3 to 0.5 for carrot. Oh, let's just go and write it down here. I don't know why that won't write properly. Maybe because there's a table there. Okay, so the intercept, or you could say the percentage change is 0 at 0.3 to 0.5 moles per decimeter cubed for courgette. I don't think that's how you spell courgette. You. And potato. No, and carrot. It was lower at what was it for carrot? 0.1, 0.1 to 1 was it? For the potato? 0.1 to 0.3. 0.1 to 0.3 for the potato. Okay, so it asks for the highest concentration of sucrose, and the intercept is higher for courgette and carrot because it was when the concentration is at, should we say, a concentration of 0.3 to 0.5 mole per decimeter cubed for courgette and carrot, but it was lower at 0.1 to 0.3 for the potato. Okay, so that's our first mark. The second thing that we can say is that if we go up, if we can trying to choose between courgette and carrot, if we look at the numbers on here, if we compare the mass gain, okay, we had the highest value for carrot. Sorry, we had the highest value for courgette. It was higher. The highest mass gain at 0 was higher for courgette than it was for carrot. Okay, so you could do that comparison to say, actually, I think courgette because this value is higher. If we compare this one here as well, this is higher for courgette and lower for carrot, so maybe that's another argument for courgette. If we compare it at 0.5 this time, this one is lower, this one is higher. So all three of those values, when we compare, let's say A with A, then B with B, and then C with C, it's always higher for the courgette than the carrot. So probably if we were trying to choose between the courgette and the carrot, we'd probably say courgette. And you can see when they then plotted it on the graph on the mark scheme, they came to the conclusion that it would have been the courgette if you'd drawn the graph as well. You might have tried to extrapolate the numbers, kind of extend the numbers and work out what it would have been in that gap, and you would have probably come to that conclusion for that as well. So the courgette had higher values, higher percentage changes at sucrose concentrations of 0, 0.1 and 0.3 moles per decimeter cubed. Okay, so that would be one mark. That would be one, that would be one, that would be one. Okay, because you compared each one. Next one. The apparatus shown in figure 16 can be used to demonstrate osmosis. So we call this visking tubing. Basically, it looks like a sort of a plastic bag, but if you zoom in, the walls of it are partially permeable. They got like little gaps. When the capillary tube with the visking tubing bag was placed in the solution Y, so a capillary tube is just a really thin tube, the level of the solution X inside the capillary tube rose from 10.5 millimeters to 26.5. So it was at the level here, okay, and that level went up. The ruler used to measure the distance along the capillary tube was accurate to the nearest 0.5 millimeters. Calculate the percentage uncertainty. So this is a skill we need to learn how to do. So calculating percentage uncertainty. So we should get an answer of 6.25. So first of all, we need to work out how much it went up by. So to work out how much it went up by, we could do 26.5 minus 10.5. And then you need to multiply that by 2 times the uncertainty, because you're measuring it once there and you're measuring it once there. So you might have made a little bit of mistake, a little bit of uncertainty about this measurement, and you might have a bit of uncertainty about that measurement. So we times it by 2 times the uncertainty, which they've told us is 0.5. They can measure to the nearest 0.5. That's what that means. Okay, and then sorry, my mistake. I should divide it by that, and then you times it by 100. Okay, and that's how you work out percentage uncertainty. So you'll be given the numbers, and you just have to do that same equation every time. What conclusions can be drawn about the composition of solutions X and Y? So we know that the liquid went up. So we know that water went from Y to X. Yeah, so if we know that the water moved by osmosis from Y to X, we must have higher water potential on this side and a lower water potential on that side. Okay, so the solution in Y outside the bag, where was Y? Let me double check before I say it out loud. Oh, yeah. So the water must have gone from Y into X. So it must have gone from a higher water potential here to a lower water potential here, because water always moves from a high water potential to a lower water potential. 6. A student carried out an investigation into the effect of ethanol on the permeability of cell membranes and meat groups. So I said to you, didn't I? Sometimes they ask you about temperature and sometimes they ask you about concentration of ethanol or alcohol. The student's method comprised of the following 5 steps. Cut equal sized pieces of beetroot using a cork borer, wash the pieces in running water, place them in different concentrations of ethanol. After 5 minutes, remove them from the ethanol, place them in the colorimeter to collect quantitative data. Each step in the student's method relies on certain assumptions. For each assumption listed below, select the numbered step from the student method that relies on this assumption. So the first assumption was the pigment will only leak into the solution if membranes are disrupted. So assumption A relates to step, and then you just need to choose one of those steps from the instructions. Assumption B, absorbance is proportional to concentration of pigment. And finally, pigment will be released when the beetroot is sliced. Okay. So first one, pigment will only leak into the solution if the membranes are disrupted. So they put them in the different concentrations of ethanol, didn't they? Because they were hoping that if the ethanol would disrupt the membrane, and pigment would leak out. So it was relying on this assumption here. Yeah, the pigment will only leak out if the membranes are disrupted. It was relying on the fact that if we had 0% alcohol, there'd be no disruption and no leakage. And then it was, as you increase the concentration, more membrane disruption, more leakage. The next one was absorbance is proportional to the concentration of the pigment. So that's relating to the colorimeter, isn't it? So that must be number 5. It's a nice easy one. That's how the colorimeter works. And the final one, pigment will be released when it's sliced. So just looking, where are they slicing the beetroot? So they cut the equal bits pieces here. So that's going to release some pigment, isn't it? They're going to accidentally cut some of the cells. So then they washed it off to wash pigment released by cutting, because they only want to measure the pigment that's released because of the ethanol. So that's why they washed it off first. So that one must be number 2. Next page. The student kept the ethanol solutions at a constant temperature. State 2 other variables which need to be controlled in this investigation to ensure it's valid. So valid means you change one variable, you keep everything else the same. So we know that they're keeping the temperature the same. There's loads of other things that they keep the same. Main ones would be surface area, cutting them to all the same sizes, so the surface area is the same. The next key one is that you use the same part of the beetroot. Can you see how similar these questions are? We talked about same bit of potato, didn't we? Same part of the potato earlier, same part of the beetroot. You could have said the time taken to wash the slices, the volume removed from the solution, the pH, the same colorimeter filter, the same blank in the colorimeter, the same variety or age or colour of beetroots. Loads of things you could say for that. Figure 20.1 shows the graph plotted by the student. Make 3 criticisms of the way the student had displayed these results. So sometimes you have to draw a graph, sometimes you have to describe the graph you draw, and sometimes you have to criticise another graph. So hopefully when you did this, you didn't plot it quite here like this. So first of all, we could have said that you would have plotted it as a line graph. So a bar graph is not appropriate for continuous data. Bar chart, would normally say. Okay, next thing is if we look at the x-axis, there's missing units. Yeah, we've got the units on the y, but we haven't got the units on the x. So we could have added the units on there. Look at the scale. 0 doesn't look like it's in the right place, does it? 0 should be just there. 0.0, so 0.0, 0.1, 0.2, 0.3, 0.4, 0.5. And then missing 0.6. It doesn't have a title. Absorbance of what? Got the units. 1, 2, 3, 4, 5, 6. Numbers look okay. Up the side. So it should be a line graph, obviously. Yeah, that's good. So quite straightforward. I think sometimes criticising someone else's is easier than drawing your own. Explain how carrying out replicates would improve this investigation. So this is something we just need to learn. So I always say that repeats makes it reliable. Okay. So why do we carry out repeats? So anything to do with repeats, we always do 3 repeats so that we can calculate a mean, so that we can identify anomalies. If we do it 3 times, we can see if one of them is really different to the others. Okay, it improves repeatability. If you do it, it's repeatable. If someone else does a different experiment and gets the same results, it's reproducible. So this, when you do it, it's repeatable. Okay, repeatability. Okay, and allows you to do statistics. That is always going to be the same list. All right, so just learn that. Don't start thinking that different words mean the same thing. Okay, those words that I've used are very specific and you need to use those exact words. Things like accurate, precise, valid, etc. have got nothing to do with it. Number 7, Beta vulgaris or beetroot is a species of beet plant. The membrane-bound vacuole of beetroot cells contains the red pigment betulin. So we've drawn that before. So wall, cell surface membrane, vacuole, and it's in the vacuole that we've got our pigment. Yeah. Beetroot was used by a group of students to investigate the effect of pH. This time they've done pH. So we've had 3 versions of this experiment. One with temperature, one with, what was it? Alcohol concentration, and now we're looking at pH. On the permeability of cell membranes. When the vacuole membrane is damaged, its permeability to betulin increases and betulin leaks out into the surrounding solution. The students cut discs of beetroot. They placed them in test tubes containing solutions of different pH for 10 minutes. Okay, so pH is their independent variable. They used a colorimeter with a blue filter to measure the absorbance. That's their dependent variable in the resulting solutions. During the investigation, the same volume of solution was used in each tube, so that must be a control variable. So 2 other variables that must be controlled in this investigation and how they could be controlled. So this specifically asks you to describe how they're controlled, but we've had similar questions to this a few times. Hopefully you're getting the hang of it. So we could say that the discs need to have the same surface area, and the way we could do that is we could use a cork borer to get the same diameter, and then we could use a ruler to measure the length. So we've described how we would make sure they have the same surface area. We could talk about controlling the temperature, and the way we would do that is using a water bath. We could say that we need discs that are the same colour, and that's by using the same beetroot for each one. Don't cut one cylinder from one beetroot and another cylinder from another beetroot because it might have a different colour. The students use their data to plot an appropriate graph. Oh, sorry, where have I gone? Shown in figure 22. The students were told that the greater the absorption of light, the greater the concentration of betulin in the solution. Describe and explain the results shown in figure 22. So describe and explain mean 2 different things. So first of all, let's have a look at describe. Always start with your x-axis and then your y-axis. So we could say as the pH increases, the absorbance decreases. We could say that it decreased quickly from, I don't know what the numbers would be, down to there, maybe pH 2 to 4, and it came down from 0.5 to, I can't read it, 0.18 arbitrary units. Okay, and then it kind of almost plateaus. From, oops, let's go down a little bit more. Plateaus, or it's a very slow decrease from pH. See, I'm quoting the numbers. I try and get into that. pH 4 to pH 12, when it went from 0.1 to 0.01 arbitrary units. Try and read them as accurately as you can, but it's not much of a scale on there. So that's your describe, something like that. Your explain is why did it do that? Okay, why did it do that? Use your science. Why did it do it? So that's your describe. This is now going to be your explain. So you could say at low pH values is more acidic, which means that you've got more H plus ions, or protons you can call them. Okay, what the H plus ions do is they denature the proteins. So then the membrane is disrupted. The reason they do that is because they form bonds, don't they? The positive charges of the ions attract the negative charges in the polypeptide of the protein, and they alter the tertiary structure, alter the shape. Okay, disrupted. Okay, so let's add the detail here. So you could say affect ionic bonds, because it's the ionic bonds where charges attract to one another. Okay, so it might be that parts of the polypeptide form a bond with the H plus ions rather than with another part of the polypeptide, which was holding the shape. Okay, so therefore, permeability increases. So more betylene released. Okay, next one, we're nearly there. This document has the mark scheme underneath. That's why it looks like there's loads left. It looks like we're only halfway. We're not, I promise. A student investigated the effect of temperature on the rate of diffusion of chloride ions from carrot cells. This is the student's method for the preliminary experiment. Cut thin sections of carrot. Place the sections of carrot in a boiling tube of distilled water. So here's our carrot. Water. Carrot. Remove the sample of water from the boiling tube and add silver nitrate solution. The silver nitrate solution reacts with the chloride ions to produce a white precipitate. Measure the absorbance of the sample in a colorimeter. Higher chloride ion concentrations will produce more white precipitate, which will increase absorbance. All equipment and sections of carrot will wash with distilled water before use. Explain why. So we only want to be measuring the chloride ions. That were produced in this part here. Okay, we don't want to be recording any chloride ions that were already there. So we want to remove chloride ions already there. And also remove any other chemicals that may react with that silver nitrate. Okay, you only needed to say one of those. It was only one mark. You could also say if it's contaminated, it would reduce the validity, because remember, validity, we only want to change one thing. If there was anything else in there, you're changing that as well. So it would reduce the validity of results. So contamination would reduce the validity of results would be a good thing to say as well. Next one, the absorbance value obtained in step 4 was used as a reference value for further tests. This absorbance value was considered to represent the highest chloride ion concentration that could be measured in the boiling tube solution. Explain why. Okay, so because if we go back to the method, in their preliminary one, they did 100 degrees. Okay, so that one would be like their highest one, and then they were going to investigate. If we go up a little bit more, they're going to investigate the effect of temperature. Okay, now temperature of 100 is likely to be the highest one. You know, if we're looking at living cells, they're not used to living at 100 degrees. So 100 is probably their highest one. Okay, so 100 degrees C is very high. So all the carrot membranes will have been broken. Down or disrupted, and therefore all the chloride ions from the cell will have been released. You only need one of those, one mark question. All right. Using the sample produced from step 3 in the preliminary experiment, the student carried out a serial dilution that produced the results in table 2.1. Hopefully we've done serial dilutions. So what you would do is you'd have your first tube, which would have 1,000. Now this serial dilution isn't halving. Okay, be careful. This one is just a tenth. So what they would use then to make it just a tenth is they'd use 9 parts water to 1 part of the tube before. Okay, so if they're going down then to 10, they use 9 parts water to 1 part from the tube before. And they're going down to 1, 9 parts water, 1 part from the tube before, and 0.1, 9 parts water, 1. So you might have done it in halves before, where you did half water, half the one before, but that would have gone from 1,000 to 500 to 250. That would half it each time. So this one is a slight different variation of a serial dilution to what you might have done in the past. Look at the trend in the results in table 2.1. Identify the anomalous result in this trend and give its expected absorbance reading. So we're looking for the trend. So we got 0.8, 0.4, 0.2, 0.1, but it's 1.8, which is actually more like 0.2. Yeah, and then we'd expect that to be 0.5, which seems fine. Okay, so it's going to be 0.18. We'd expect it to be 0.10 expected. All right. Doesn't quite follow the pattern. Oops, where have I gone? Yeah, done. The student used the method from the preliminary experiment to plan their investigation into the effect of temperature on the rate of chloride ion diffusion. Describe 3 variables that the student would need to control when planning this investigation. Isn't this familiar? Include reasons why each of your chosen variables must be controlled. So we could have said the volume of distilled water. Why? Because a larger volume of water would mean a lower concentration of chloride ions, okay, or of the precipitate that it makes. We should have controlled the volume of distilled water removed. We could have said in boiling tube there. Why? Because a larger volume removed will increase the number of chloride ions, or increase the precipitate formed, or increase the absorbance. We could have talked about the time. So we should have left it for the same amount of time in water bath. Okay, so either we should allow it to reach desired temperature because, oh, sorry, yeah, that is it. Or if you didn't have enough time, then not as many chloride ions will have had time to diffuse across the membrane. You could have explained it in that way. We could have said the surface area of the pieces of carrot. Because larger surface area would increase diffusion rate. So more ions would have passed across the membrane. Or we could have said the number of drops of silver nitrate. I'm not going to write them all down because a larger concentration would increase the absorbance because it would increase the precipitate formed. Or we could have said we should have used the same filter in the colorimeter because the filter is changes the wavelength that reaches the sample. You could have said the same reference solution in the colorimeter, so the same blank, like the same water equivalent, prevents zeroing errors, or you could have said same part of the carrot tissue or taken from the same carrot, because different carrots will have different chloride ion concentrations. And that, I think, is the end of the paper. Well done if you made it to the end. Brilliant.